Welcome back to my channel Adhyan with Flow. Okay, let's discuss about DNA versus RNA. First of all, you have to know about DNA. I already described all things that what is DNA. Draw a line, center line, okay, and then differentiate. But here is space is less na. That's why I'm giving you properties one by one. DNA is what? Write down full form. Deoxy ribose nucleic acid. Deoxy means, wait, what you understand by deoxy? Guys, here they contain only one oxygen, 1 2 3. At the position of 2 they does not have extra OH group. That's why we call it as a deoxy ribose nucleic acid, because this is a sugar group. We can say it as a ribose. Guys, next differentiation is what? Next property is what? You have to write down that they are made up with three parts: protein, sugar and nitrogen base. Then you have to write down that nitrogen bases are in two parts: purine and pyrimidine. Purine is what? Adenine and guanine, and pyrimidine is what? Cytosine and thymine. Next property is what? Adenine pair with thiamine. This is the purine part and this is the pyrimidine part. This is the double bond, double hydrogen bond. Another property is what? G and C pair with 3 hydrogen bond. Clear or not, guys? Okay, next property is what? Guys, I'm writing here properties, okay? Next property is what? The DNA is what? DNA is what? Double helical structure. Double strand structure means they present in double helical form. Yes or not? Now DNAs take participation in where? Replication means they can amplify their copy, means this strand will split and another strand will form 2 to make another 4. Each split and make another copy, means 2 make 4, 4 make 8, 16 and further on. Now guys, next thing is what? Location. Where DNA is found? Nucleus. Where nucleus is present? Cell. Clear or not, guys? Tell me, clear or not? So these are the properties of DNA. Many scientists prove that DNA is genetic material. Many scientists prove this thing that DNA is genetic material. Now next property is what? They present in complementary form. Complementary form means 2 strand, 5 prime to 3 prime, 5 prime to 3 prime. Complementary to each other. If one strand goes 5 prime to 3 prime in direction, then another strand goes toward the 5 prime to 3 prime in direction. Let me show you. Here is what? Double strand structure, 5 prime to 3 prime, and another is what? 5 prime, complementary. Here is 5 prime, here is 3 prime, here is 3 prime, here is 5 prime. Look at here, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Look at here now, phosphate and sugar. And this is orientation same but opposite in direction. Phosphate and sugar, phosphate and sugar, phosphate and sugar, phosphate and sugar. Now next thing is what? G and C pair with 3 bonds, A and T pair with 2 bonds. I am gonna giving you numbering here 1, 2, 3, 4 and this is 5. That's why this is the 5 prime. I am giving you numbering, the end thing is 1, 2, look at here 3, this is the 3 prime, this is a free end. It's a long chain. They take only 4 nucleotide but there is long, okay. Now here is what? 5. So this is the 4, 3, 2, 1. 5 na? This is the 5 na? 4, 3, 2, 1. Understand? Now how they are 5 and how this is the 3? 1, 1, 2, 3. Clear or not? Double bond clear or not? Triple bond clear or not? They are complementary or not? Yes, they have phosphate, sugar and nitrogen base. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen. You have to remember this structure. Look at here. This is the simple diagrammatic form of DNA. Double helical structure. And here is what? Nitrogen base. And phosphate plus sugar make their backbone. This is the backbone. Clear? Okay, now let's move towards the RNA. Now, what is the special properties about RNA? Full form is what? Ribose nucleic acid. Ribose nucleic acid means, because this is not deoxy, it means they have at the both end OH group. That's why we call it as a ribose. Now this is not a genetic material. This is not a genetic material, but yeah, they work as a genetic material in primitive organism like bacteria, virus. Okay. Now next thing is what? They are single strand structure. Yeah, guys, They are single strand structure, and they made up with what? They made up with what? Phosphate, same, sugar, and same nitrogen base. Now nitrogen base is what? Purine and pyrimidine. Purine is what? A and G. Pyrimidine was C, but at the place of thiamine, uracil is present. In the case of DNA, thiamine is present, but in the case of RNA, uracil is present. Now, guys, RNA take participation in translation because DNA replicate, make a multiple copies, but RNA can't make their multiple copies. You have to write down. They can't replicate because this is a single structure, yeah. So they make their multiple copies here and here. Now each strand of DNA take participation on transcription, and they make what? And they make RNA. RNA, what he do? He make protein. So RNA take participation in translation. With the help of translation, what they do? They make protein. Where RNA will found? In cell. In the cell, RNA will found. And many types of RNA is present here. mRNA, tRNA, rRNA. mRNA work is a messenger RNA, tRNA work is a transporter RNA, rRNA is what? Ribosomal RNA. So these are the properties of RNA. So you can differentiate DNA and RNA, yes or not? Let's begin with what? Let's begin with replication. What you understand by replication? I already describe you everything, guys. What is replication? This is the DNA. This DNA is split, and each DNA make their own copy. This is the procedure of replication. But this is not an easy process. No, guys, this is not that much easy. No, this is not that much easy. Assume, guys, this is the what? This is the what? DNA. They are paired with nitrogen base with the help of hydrogen bond. Now what kind of enzyme come and they take participation in replication? Now this is the double stranded DNA. Are you got it, madam? This first step is clear or not? Tell me. This first step clear or not? 5 prime to assume 3 prime and 3 prime to 5 prime. Are you understand this thing? Now guys, what happened with this? First helicase come and break hydrogen bond. Helicase come and break hydrogen bond between adenine and thymine, between guanine and cytosine. Now there are hydrogen bond. This is cut by helicase. So helicase come happy and they cut these hydrogen bond. Cutting. So this kind of orientation will present. Yes or not? Still they are not cutting. Ligase is continue forward, but after cutting these strands 5 prime to 3 prime, 5 prime to 3 prime. After cutting this fragment, now what happened with them? What happened? Next protein, SSB protein, come and attach here. So they can't easily combine. If they attach now, they can't combine. If this SSB protein, single stranded binding protein, is not present now, so they can easily attach towards further. But with the presence of SSB, they can't attach. Now second step is what? SSB protein come and bind with their DNA single stranded. Now third step is what? What happen? Now primer will come, not girls cosmetic primer. Primer will come and attach. Primer will come and attach at 3 prime end. So here primer will attached and here is 3 prime end. Now here primer will attach. Let me change pen. Primer, here is primer. He needs 3 prime end. Always he needs 3 prime ends because 3 prime end have OH group. That's why he needs 3 prime end. Now here is primer is attached. Primer is attached. Now fourth step is what? Fourth is what? DNA polymerase will come. DNA polymerase will come and attach on primer. DNA polymerase come. DNA polymerase will come. And what they do? They attach what? Nucleotides. Nucleotides made up, I told you na, nucleotide made up with what? Phosphate, sugar and nitrogen base. Then why you? Then nucleotide will come and attach with these strand. nucleotide will come and attach with these strand, nucleotide will come and attach with these strand. But case of 3' end, here is what? They can't synthesize their self. They can't synthesize their self properly, but they can easily synthesize. Because guys, they move in this direction, 5' to 3'. I told you guys always complementary. This strand is 5 to 3' now, so here is 5' to 3'. They continue in this direction. But what's wrong with this primer and DNA polymerase? What? They want to move in this direction. They want to move in this direction, but there is some breakage, because this is the 5' to 3' now. So this is the 5' to 3'. First, okay, first nucleotide will complete. Okay, assume this first nucleotide is complete. Now what happen next? Because helicase break other hydrogen bond. So now what happen? I'm drawing here 3', 5', 5'. Here is primer, here is DNA polymerase, and they continue make strand. But what's wrong with another when helicase move forward? But here 5' to 3', 5' to 3' in this direction. First thing is complete, 5', 3'. Now here in this direction this is complete. But now what happened with these things? Now what happened with these three? Now again another primer will come and attach here, and they synthesize in this direction with the help of DNA polymerase. So now this is the non-stop continuous strand. We can call it as a leading strand, and this is what? Lagging strand. This is the continuous, this direction, no need of other primer, but they need when helicase cut, because in helicase again he cut this strand. Now what happened with this strand? Now what happened with this strand? This strand is continue 5' to 3' direction, but they are break. Then this, then again after cutting hydrogen bond, again this. So there is breakage. This is the continuous, but this is the discontinuous. This is the parental, but this is the new strand and this is the new strand. Now what happened with these fragment? We call it as a Okazaki fragment, and they further attach by ligase. So in future we take this strand. All right. So that's all about DNA replication, guys. DNA replication I completed. Okay, I'll make, okay, wow. Okay, again you can understand betterly. This is the DNA. This is the what? DNA. Now what's wrong with DNA? Step 1 is what? Helicase come and cut these strand. Here is double strand now, guys. Here is hydrogen bonding. Helicase will come and move in this direction, and they break. So they split in this manner. Now what happened with this? Primer will attach, DNA polymerase attach, and they make nucleotide. They add nucleotide continuous. But in this, here to here, then form, then here to here, then here to here, because they are continue. Helicase is cutting this thing, and they continue synthesize. But here, when they break, then again they form. Again they break, again they form. Again they break, again they form. Again they break, again they form. So step 1 is what? DNA. Then DNA split by helicase. Now this is parental, Now what happen? SSB come, single strand binding protein come and attach to split strands. Now what happen next? Now primer come, bind at 3' end of parental strand. Here now this is the parental strand, and here 3' and the primer will come. Now after primer what happen? DNA polymerase come and attach what? Nucleotide, and nucleotide made up with phosphorus, sugar and nitrogen base. So two types of strand will form, leading and lagging. Leading is what? Continuous strand, and lagging was discontinuous strand. They synthesize non-stop 5' to 3'. Okay, they are discontinuous, so they produce Okazaki fragment. Leading strand does not need ligase enzyme, but they need ligase enzyme. Ligase is what work as a glue. They attach this thing, they attach this thing, they attach this thing. Now two strand will form, parental and leading strand, and here is what parental and lagging strand. So that's all for now, guys. Here is your DNA replication is complete. You have to learn and read your book. That's all for now. In our next video, we will cover transcription, how DNA split and they form what? mRNA. Means DNA to RNA. We can say mRNA. Okay, guys. So keep smiling. We will meet in our next video. And don't forget to subscribe my channel. Keep supporting me, guys. Please. Adhyan with flow. Thank you so much.